Hello everyone, and welcome to our Discovery Series webinar. My name is Emma, and on behalf of Ochre Global, I'd like to thank you all for joining us today. Today, we have three fantastic presentations surrounding the topic of novel strategies to accelerate discovery of translatable therapeutic targets. This webinar will feature presentations from Zoltan Spiro, Target Discovery Lead at Turbine AI, Steve Harrison, Chief Scientific Officer at Engine Bio, and Quin Wills, Chief Scientific Officer at Ochre Bio. We are also joined by Dave Madge, Vice President at WuXi AppTec, who will be introducing the session and moderating our Q&A. Before we get started, I'd like to share a few words about WuXi AppTec, who are kindly sponsoring today's webinar. WuXi AppTec provides a broad portfolio of R&D and manufacturing services that enable companies in the pharmaceutical, biotech, and medical device industries worldwide to advance discoveries and deliver groundbreaking treatments to patients. With industry-leading capabilities such as R&D and manufacturing for small molecule drugs, cell and gene therapies, and testing for medical devices, WuXi AppTec's open access platform is enabling more than 5,200 collaborators from over 30 countries to improve the health of those in need and to realize our vision that every drug can be made and every disease can be treated. Finally, a few aspects for our delegates to note. Following the three presentations, we will be hosting a live Q&A with all speakers. Please submit your questions through the chat feature at the bottom of your menu display. The full recording of the webinar will be made available to download on our secured website, and the details will be sent to you after the webinar. Without any more delays, I'd like to hand over to Dave to introduce the session. Dave, over to you.
Hi everyone, thanks for joining this webinar. Today we're going to take a look at recent developments in target identification strategies in drug discovery. My name's Dave Madge and I look after the EU business for WuXi AppTec. During this webinar, we're going to showcase some of the latest technologies and platforms for identification of novel disease targets. And we're going to look at the use of disease simulations, deep phenotyping, combinatorial gene editing, and applications of artificial intelligence. I'm very happy to introduce three guest speakers to cover these topics from the perspective of their respective organizations. Zoltan Spiro is from Turbine AI, Steve Harrison is from Engine Bio, and Quin Wills is from Ochre Bio. Each of these companies have their own strategy for target identification, and we're looking forward to hearing from them over the next 45 minutes or so. To set the scene a little, it's worth remembering that existing drugs are engaging with only an exceedingly small fraction of the human genome, with only around 700 proteins targeted by approved drugs. However, this is beginning to change as we develop a deeper understanding of more diverse opportunities for disease intervention, such as control of transcription, translation, targeted protein degradation, control of post-translational modifications, and many other strategies. Indeed, even amongst the gene families we have shown to be druggable, most are underrepresented in terms of molecules getting as far as the clinic, the blue portion of the bar charts on the left here, with a few more having identified small molecule ligands, the green portion, but the majority in red having no known small molecule ligands. Furthermore, we also know that the majority of monogenetic disorders have not yet been drugged, as shown in the right-hand panel here, with around 6,000 proteins associated with at least one rare disease. To help us modulate more diverse targets, there's been rapid progress in recent years towards validating new modes of action, as well as the creation of ever more inventive types of molecules to address these modes of action. We can now engage with the up and down regulation of RNA, for example, as well as stabilizing or disrupting protein-protein interactions and repurposing housekeeping processes to enhance the degradation of target proteins. Whilst the scope of application of small molecules remains broad, there are modes of action where antibodies, protein replacement, oligonucleotides, or cell and gene therapy provide a more tractable strategy. So there are lots of potential drug target opportunities that we have not yet exploited, but perhaps could now using alternative strategies. Picking amongst those targets to find those that offer the most disease-relevant and tractable opportunities is the subject of this webinar. I hope that sets the scene for Zoltan, Steve, and Quin, and I hope you enjoy their presentations. I also hope you can join us for a short Q&A session after these presentations. Thank you very much.
Thank you very much for that introduction, Dave. And we'll now get started with our first speaker, Zoltan Spiro, Target Discovery Lead at Turbine AI. Zoltan's talk is entitled Computers Take the Lead in Oncology, Simulation-Driven Discovery of Translatable Targets. Over to you, Zoltan.
Hi, everybody. My name is Zoltan Spiro. Welcome to this webinar. Today, I will be telling you about how we at Turbine AI simulate a cell and how do we use these simulations, the computer simulations, to identify novel drug targets in oncology. In Turbine, we built a simulated cell. This simulated cell contains nodes, which are proteins and their interactions. So it's a protein-protein interaction network. And we are using transcriptomic and genomics data to model existing in vitro cell lines. Thereby, we have around 800 in silico cell lines in our network. These cells are viable in silico in our simulations, but we can perturb the system. And by these perturbations, we are able to read out what conditions cause cell death in a given cell type. So we are using these in silico cell lines to perform these simulations in order to identify novel drug targets that kill cells in a context-dependent manner. We are then using these hypotheses coming from the simulations, and we validate them in vitro. And there, at the end of the validation, we have our targets, a biomarker, and we are also able to do drug combination simulations. A very important aspect of the Turbine workflow is the learning loop. We are using the in vitro generated data to feed back to the model and to train the model to become even more predictive. The very big advantage of in silico simulations is that it takes much less time than in vitro CRISPR screens. So basically, the whole process from running the screen, doing the simulations, translating the results, and validating the targets in vitro takes 11 months. After these 11 months, we are then able to start the drug discovery program. First, we were using our platform, and we were testing our platform with the big pharma companies. We were helping their drug discovery programs, identifying novel biomarkers for their existing drugs. In the last couple of years, we started using AI to make the network even more predictive. And in the last 2 years, we became a drug development company, and we already identified 8 novel target biomarker pairs, which are our pipeline at the moment. You can see that 2 of our targets are already in the drug discovery phase. Some are in the target validation phase, and some are now being evaluated in silico, looking at the data, and we are deciding whether we would be progressing them to target validation. Today, I would like to talk about the Insight 1 screen, which yielded 4 target candidates. And in the rest of the presentation, you will learn how we run the screen and what were our criteria for target selection and how we are now validating the results with WuXi. So first, generally, I wanted to show you what kind of perturbations we are able to perform in our system. Here, you have a representation of a protein-protein interaction network. You have the interactions between these different proteins. This can be negative, this can be positive. There's also strength of the interactions depending on their nature. We can remove individual nodes, individual proteins, which resembles a situation of a CRISPR knockout. And thereby we can look and check what kind of phenotypes the different cell lines in silico give upon the perturbation of that one given node. We are also able to use a fictive inhibitor, and by applying this fictive inhibitor at different doses, we are able to see which cell lines are more sensitive to the inhibition of this given protein, which are more resistant, which somewhat mimics the effect of a drug. And it's very useful to really try to understand why a given protein inhibition gives rise to sensitivity in one type of cells and why it doesn't in another type. This is the perturbation that we were using in the Insight 1 screen. However, I also wanted to tell you about the edge perturbation option. So we can basically switch off distinct interactions of a given protein. And this is resembling the situation of domain-specific inhibition. And we can get more insight on the mechanisms, how a given target works. And we are also able to combine the target inhibition with drug treatment, looking into existing drugs, finding out different biomarkers, or also using our own drugs in the future to refine our targeting hypothesis. So the Insight 1 screen was run this year in March here at Turbine. We were focusing on indications with high unmet medical need. Based on the high unmet medical needs, we were looking into what kind of in silico cell models we have. And we were focusing our screen on pathways that we think are modeled in a reliable way. So at the end, we ran this screen on almost 500 in silico cell lines, and it yielded very interesting results. We started with 1,500 proteins. This is not all the proteins that are in the network. We selected these because we felt confident about the modeling aspects of these proteins. And then we were using these 1,500 proteins, and we went through a filtering step, a filtering workflow to arrive to our shortlist. Our selection criteria included the in silico effect, and this is, of course, something that you're not able to do without such a simulated cell model. We were looking into targets which are not yet in clinical development. We wanted to remove targets which have toxicity issues or they are common essential. We were also looking into targets which are expressed in tumor tissues, and we wanted to make sure that the targets that we progress, they possess low to medium competition. From these 42 target candidates, we were then performing something that we call target translation, where we went into each and every target. We then looked into its novelty. We were also looking into in silico pathway activity readout, which I will be showing you to you in the next slides. And we wanted to progress only targets where we were sure to understand why sensitive cells die and why resistant cells don't die. We were looking into competitive landscape. And obviously, we don't only want to identify targets which are biologically interesting, but we want to identify targets which also have higher chances of being druggable. So these are the kind of mechanistic readouts that we can generate following an inhibition of a target. Here I'm showing you a mode of action plot with the pathway level readouts. So here, each column represents one signaling pathways. The red color indicates the downregulation of a given pathway, and green indicates upregulation. This plot is divided into two parts. In the upper part, you see the pathway activities of sensitive cell lines, and in the lower part, you see pathway activities of cell lines which are not sensitive to the inhibition of a given target. And we are looking into signaling pathways where basically we have a clue, we have a hint why sensitive cells die and why resistance don't. And this is exactly something that we then translate. We try to make sense of this, and down the road, we are trying to validate these hints and these predictions. Apart from inhibiting a given target, we are also using our simulated cell to identify potential biomarkers. Targets are really important and interesting if you have the patient stratification biomarkers with that. So this does make sense. And again, this is not something that you can do in vitro. So you have to use in silico simulations. Together inhibiting the target, we are able to perform loss of function studies, underexpression studies, overexpression studies, and also gain of function studies. So this means that apart from inhibiting one node, we are playing around with either the functionality or the expression levels of some other nodes in the network. And then what we are doing is we are looking into changes of sensitivity. So basically, we want to identify biomarkers which decrease the IC50 values of this fictive inhibitor, of the applied fictive inhibitor, and also increase the kill rate of the target inhibition. And then here on this graph on the right upper corner, you see those biomarkers, those perturbations, which sensitized the cells to the inhibition of a given target. Then comes a process that we call biomarker translation. We are focusing on effect strength. We are building up then the target biomarker hypothesis based on literature data as well. We are looking into patient survival curves. We then prioritize indications based on the prevalence of these different biomarkers. And by that, we have a package where we have a target, we have a biomarker, and we understand why this actual biomarker sensitizes the cells to the inhibition of this target. And thereafter, we go to WuXi and we set up a target validation experiment plan with them. And basically, we are dividing our cell lines into 3 different kind of groups. First, there's a group which we also found to be sensitive to the inhibition of the given target. And also in that map, the in vitro dependency seems to show that indeed this cell line will be dependent on the knockout of this given target. So here we expect that those pathway activity changes that I showed you before are indeed true, and the viability will be compromised. On the other hand, we choose control cells which are not dependent on that map and also not in our simulations. These are used as controls, like I said. And also, if this holds true, if these cells indeed don't really care about the absence of this protein, these cells will be used for follow-up biomarker validations. So on top of the target knockout, we also will knock out or perturb our biomarkers. And then there's a third group, which is very exciting for us, where basically we could see that that there are some cells that we seem to find sensitive, whereas in that map they weren't picked up. These are, of course, very interesting for us. And here we also predict that the pathway activity will be down and viability will be compromised. So this is what I wanted to tell you. Of course, after this target validation, we then evaluate whether the target has been indeed validated in vitro. We then use the generated data to feed it back to the model, and we progress the target candidates for biomarker validation as well as drug discovery. Thank you very much, and I'm happy to take questions.
A great talk there from Zoltan, and we'll move swiftly on to our next speaker, Steve Harrison, Chief Scientific Officer at Engine Bio, who will be speaking on integrating artificial intelligence and combinatorial CRISPR screens to identify novel molecular targets in cancer and neurodegenerative disease. Thank you, Steve.
Hi, I'm Steve Harrison with Engine Biosciences. Thanks very much to Dave and to WuXi for allowing us this opportunity to present with such an esteemed panel. So, Engine Biosciences, we use machine learning and biological experimentation, particularly high-throughput CRISPR screening, to identify molecular targets and also patient selection biomarkers. So for a bit of background, Engine was seeded in 2018, and over our lifetime, we have raised over 53 million in funding from our Series A, from the seed, and from non-dilutive financing. And some of the investors in Engine are shown on this slide. We use an approach known as synthetic lethality. Many of you will be familiar with that, in order to identify precision medicines. So specifically medicines that are targeted to particular patient populations by virtue of the presence of biomarkers in their tumors. it's really important to identify additional therapies in this area because over 90% of cancer patients still lack approved precision medicine options. A classic example of a precision medicine is the BRCA-PARP approach, in which PARP inhibitors will only kill cells in which there is a mutation in the BRCA gene. And so tumors that are mutant in BRCA will be killed by PARP inhibitors, but normal cells in those patients will be unaffected, and thus giving a good therapeutic index. Our approach to identifying such synthetic lethals and precision medicines is by virtue of a combination of computational approaches, whereby we screen the large amount of omics data that has been generated by virtue of many clinical trials now in which DNA sequence and RNA transcriptomic data has been collected. We then apply our machine learning algorithms. We identify across the entire genome synthetic lethal pairs, which we can then test in our high throughput CRISPR-based validation approach. We can test tens of thousands of hypotheses for good biomarker target combinations. And then when we validate those and prioritize those, we enter into internal drug development activities for the pursuing the most interesting of those pairs. So this is how this approach works. We call the platform NetMAPPR. As I mentioned, we access data from both proprietary sources, so clinical data that we have specifically acquired, as well as the public data like TCGA from the Broad Institute. We then train our machine learning algorithms and to interrogate that information. And if you think about it, there are 20,000 genes in the genome. So that allows for 400 million possible pairs of genes. So that's actually 200 million pairs of biomarker with potential target genes. So that's a huge basis for an initial screen. It allows for unbiased identification of pairs. It allows us to make many hypotheses about good biomarker target combinations, in fact tens and hundreds of thousands, which we can then test in tumor cells, human tumor cells, using our high throughput combinatorial CRISPR screening approach known as CombiGEM. And out of those screens, we identify synthetic lethal pairs in the order of tens now, which we then go through extensive validation and identify top priority pairs that we move into drug discovery. And the result of that, we have now a growing pipeline. In particular, our lead program 812 is targeting liver, ovarian, and colorectal cancer. It's a kinase target. And with an associated biomarker. We also have a number of other screens ongoing, which have yielded additional programs in the pipeline, as well importantly as identification of biomarkers for known drugs. And I'll talk a little bit more about that in subsequent slides. In addition to the work in oncology, we also have earlier efforts in other therapeutic areas, including dermatology, in which we've had a successful collaboration with Procter & Gamble, as well as work in screening in neurodegeneration to identify targets for areas of high unmet medical need. So a word about the algorithms that we use for target identification and biomarker identification. We use machine learning algorithms which are trained on curated data sets. So we have the largest curated data set of synthetic lethals, over 10,000 synthetic lethals in our NASCAR database that have been accumulated from multiple different sources, including the literature. We use that to train our machine learning algorithms, one of which we refer to as Block. This is a model that has over 50 features and relies on 1 million data points. And we use that to interrogate the entire genome for hypotheses for good biomarker target combinations. We also have another algorithm referred to as Charger, which is a functional genomics data mining approach looking at cell line multiomics and loss of function screen data, as well as additional algorithms, including our ABC algorithms. These all combine to generate multiple hypotheses for potential synthetic lethal interactions, which we then test at high throughput in combinatorial CRISPR screens. And the approach that we take is a little different from screens that have been applied by other groups. So, for example, the non-isogenic screens applied by the Broad Institute and by KSQ, which do genome-wide screens across large panels of cell lines, look for those gene knockouts that end up killing cells, and identify which cells are killed, and thereby infer from the genetic backgrounds what might be background mutations that predispose those cells to killing. It's a little messy. There are highly variable genetic backgrounds there. So an approach that has been used to make that a little cleaner would be to use isogenic cell line pairs for particular biomarkers, either cells that either plus or minus the biomarker, and then to do genome-wide screens in the presence of those cell lines. It is a cleaner approach. It's a little laborious because you have to generate isogenic cell line pairs for each of those biomarkers. The approach that we apply is to do a combinatorial screen in which we knock out both the biomarker and the target gene simultaneously. We can screen 100,000 combinations, and because we're doing this in defined cell lines, several cell lines per screen, it's well controlled in terms of genetic background. It's also pretty scalable, and we can get data out of such a screen within a couple of months. And if necessary, we can also expand the screen to knocking out more than 2 genes simultaneously. So this is an example of how we have done this. So this is related to our 812 gene pair. As I mentioned before, we looked across the entire genome using our algorithms. We identified 100,000 or so hypotheses for biomarker gene combination, target gene combinations. Then in our wet lab combinatorial screens, we validated, tested those hypotheses in our CombiGEM screen, out of which we identified 44 gene pairs that have particular interest to us. We then further narrowed that down using criteria such as the druggability of the target, the so-called gene B, and the prevalence in cancer of the biomarker, the so-called gene A, the extent to which there is a gene interaction or synthetic lethality, the extent of the killing of the combination, and also other considerations such as the development path for such a pair. Then we do further validation, in which we are looking across multiple different cancer cells to identify if the effect that we see is in fact robust and not just limited to the specific cancer cells that we originally tested. And then once we've identified high priority pairs, we test those in xenograft studies and also in proceed into drug discovery. So this is an example of some of the data that we generate in our synthetic lethality approach. So we look in validation experiments in which we are knocking down either the single biomarker gene or the single target gene, and then in combination. So in such experiments, looking at, say, the panel on the left here, we normalize viability to the viability of an unmodified cell line. So this is NTC, or non-targeting control, for the CRISPR experiment. That's a viability of 1. If you knock down an essential gene like POL2, you get complete loss of viability. We looked at a control, a known synthetic lethal pair, where you knock down MTAP as a biomarker, has relatively little effect on viability. PRMT5, the associated target with that, a bit more of effect on viability, but in combination they have a more profound effect. For our biomarker, which we call the AP gene, and our target kinase target, the 812 gene, both of those on their own have relatively little effect. That's the orange and green bars, and yet in combination in the blue bars. And that's true not only in the liver cancer cell lines that we have tested, but also in ovarian cancer cells and colorectal cancer. So this synthetic lethality has what we call penetrance. That is to say, it has a robust effect in multiple different cell types. Another thing that we can do is actually look for known therapeutics that perhaps may not have had good enough efficacy in clinical development, perhaps in phase 2, in order to identify patient populations that might have a better response. And so we show an example here for BRD inhibitors. And when we look using our computational and CombiGEM approaches, we identify a number of interesting biomarkers, one of which is shown here is the NOP5 biomarker. It shows a strong synthetic lethal relationship with BRD inhibitors, multiple BRD inhibitors, with strong statistical significance. The loss of function of NOP5 significantly increases the potency, as I'll show in the subsequent data. And it's a highly prevalent biomarker in multiple cancer types, including prostate, colorectal, and other cancer cells. So in these experiments, we're doing dose responses of BRD inhibitors, a couple are shown in the 2 different panels. In the blue lines, this is in the absence of a knockdown of the biomarker. And then in the presence of presence of the knockdown of the biomarker in these cells, you can see that that curve is shifted to the left. That is to say, much lower amounts of inhibitor are required to kill those cells. So that's a substantial increase in sensitivity of cells to the inhibitor in the absence of that biomarker. So that would be potentially an interesting population of cells to treat with these BRD inhibitors. So in summary, Engine's approach is to bring precision medicines to the clinic. We have a unique combination of machine learning and functional genomics, and allows a broad and unbiased target biomarker identification. We have a proven ability to identify precision medicine targets with the target and associated biomarker. We have the opportunity to improve the efficacy of failed agents that may have not had sufficient efficacy in clinical development by providing novel patient selection biomarkers, and we're applying in multiple therapeutic areas. So I look forward very much to the discussion, and thank you again.
Thank you very much, Steve. We will now move on to our final presentation. Quin Wills, Chief Scientific Officer at Ochre, will centre his talk around deep phenotyping to address the translatability crisis in liver disease R&D. Thank you for joining us, Quin. Over to you.
Hello, everybody. Over the next 10 minutes, I'd like to share 3 things with you. The first is the reason why Ochre exists. I'll set the scene with learnings from 2 decades of liver research, summarized here as 5 factors that I propose are limiting therapeutic innovation. And it might surprise you to hear this from a genomicist, but genomics and big computation alone are not going to save us all. So the question then is, what will? I'll attempt to answer this in one small way by describing what we're doing to better integrate advanced genomic techniques with functional human liver studies. Then I'll briefly finish off with how we are translating our findings into RNA therapies for donor livers. But first, the translatability crisis. If you're not familiar with liver disease, this plot may shock you. Chronic liver disease is the only major global killer on a steep trajectory upwards. Despite this, therapeutic options are limited. This, I would argue, is for 5 interconnected reasons. The first reason is that liver targets are a bit of a riddle wrapped in a mystery inside an enigma. Chronic liver disease struggles with the same problem faced by other chronic conditions, such as neurodegeneration. Namely, it is silent for most of its trajectory. And so there remains a fairly limited understanding of the mechanistic biologies that are playing out on a scale of decades before clinical disease presents. A second reason is that in vitro and animal models tend to be very poorly predictive of human liver disease. The generalizability of simple acute research models to the many inter-individual variations within human chronic disease trajectories can sometimes be questionable at best. Thirdly, with a lack of reliable biomarkers for much of chronic liver disease, we are forced into performing large clinical trials reliant on long-term endpoints. Large, long, and expensive clinical trials disincentivizes smaller therapeutic companies from entering the space, despite the large unmet medical need. Another reason for why innovation can prove challenging is that by the time chronic liver disease presents, it's a complex milieu of many angry cell types contributing to the problem. Now, no parent would ever hope to resolve hostility between their offspring by just telling one child off. And yet, somehow, when it comes to livers, we keep narrowly focusing on individual cell targets to shift hostile liver environments. Finally, it's important to recognize that while Big Pharma does have an interest in some aspects of chronic liver disease, that transplant medicine is too often an indication to be of interest to most, even as a segue into chronic indications. And so with disincentivized smaller companies and narrowly focused Big Pharma, the industry engine for generating new therapies isn't optimal. And so it's with these challenges in mind that Ochre Bio was founded, starting with a focus on deep phenotyping. Thanks to genome-wide association studies, chronic liver disease genetic hits now abound. Here is one of many recent studies that can be found within public domain repositories. Some of you may recognize these genes. And while the biology behind some of these genes seems clear, for others, it's not so obvious. By that, I mean, which cell type is the gene most active in, if the liver at all? At what point in the chronic trajectory is the gene relevant? And what is the basic biology that the gene is even modifying? Deep phenotyping attempts to answer these questions by connecting molecular phenotypes with tissue phenotypes and then clinical phenotypes, using new technologies such as single-cell and spatial sequencing, combined with computational modeling. It's an approach we use throughout our human liver tissue research, starting with deep phenotyping to discover new targets. Here, for example, are two H&E biopsies from a study of 1,000 transplanted livers. Each liver was imaged 6 times, covering multiple stains. We first used machine learning to standardize the calling of recognized histological features. Then we used it to classify tissue niches such as zonality or clusters of diseased cells. Here the algorithm is highlighting clusters of ballooning cells in blue. Then we spatially RNA sequenced each liver to map molecular behavior directly to local histology and 150 clinical phenotypes. For example, the top plot shows hit genes associated with the percentage lipid in the tissue, while the bottom plot is of genes associated with graft failure. In both, the x-axis is a measure of importance derived from traditional statistical modeling, whereas the y-axis is a measure of importance seen through the lens of a nonlinear machine learning model. Another approach for us is to deep phenotype liver tissue in the lab to better dissect out biology. Each one of these stacked benchtop mini bioreactors, for example, can maintain 96 slices of human livers to study what happens when we knock down a gene, thus allowing us to study liver biology at scale. At the top, I'm showing you what one of these cultured liver slices looks like. But rather than just imaging or assaying the slices, we can single-cell sequence them to once again map molecular behavior in specific cells to the phenotypes that we measure. Finally, the jewel in the crown for us is directly studying liver biology within perfused human livers, where we are able to profile both the perfusate and the liver itself. Here are 3 examples of what I mean. In this heat map, I'm showing you an example of perfusate biochemistries that differ between a steatotic liver, a fatty liver, and its matched control. Yellow means that the phenotype is greater with the steatotic liver, while blue means it's less. The x-axis shows temporal changes in 2-hour increments. Shown here is an example of where we have a subset of circulating perfusate biomarkers from a proteomic panel run on a perfused liver given an ischemic perturbation at the time point shown in red. It's a protocol developed by Dr. Hussein Abbas, pictured right, where we are detecting changes in circulating tissue remodeling, inflammatory, and cell stress proteins. But more important than the details around individual markers is what I'm trying to show at a high level, here and in the previous heat map, that it is entirely feasible to study meaningful changes in liver physiology with machine perfusion. Not only changes in biochemistry and proteins, but changes in biological pathways inferred with RNA sequencing. Here I'm showing you an example of a perfused donor liver, where within hours we can transcriptomically infer shifts in lipid metabolism, cellular stress, and inflammatory response. But here's the really exciting bit. By only working in human tissue all the way, and by sequencing all the way, we are assembling a consistent set of data at multiple scales and throughput into a single computational model. We're calling it our in silico liver, and estimate that at some point next year, we'll have generated enough sequencing data to start making reliable predictions about genes ahead of testing them. This means we don't need to compromise on speed or scale in order to study the liver at this level of complexity. The computational model constantly triages more promising genes so that we don't waste time on those that don't work. We like to think of it as getting the best out of both computational and experimental modeling. So what we're doing with all of this information is translating it into RNA therapies, beginning with RNA therapies intended for the use with machine perfusion of suboptimal donor livers. This is a fluorescence histology image from a biopsy of a human liver that was machine perfused with one of our RNAs, shown here in pink. The type of RNA we focus on is siRNA, or small inhibitory RNA, a naturally occurring gene downregulation mechanism. A chemically modified siRNA can be synthesized to only enter a specific cell type and then silence a gene of choice. A strategy we focus on for 3 reasons. The first is speed. As the world has come to realize with RNA vaccines, one can go from target to therapy in a matter of days. The second reason is that the effect of chemically modified siRNAs can last for 6 or more months. This means we can treat a liver on a machine with a single dose aimed at improving transplant outcomes, but then can also consider longer-term changes in liver physiology without the need to administer a systemic therapy. This, we hope, will allow us to perform tractable transplant trials with a keen eye on possibly expanding the therapies into patients living with chronic liver disease. Finally, it's my personal belief that the cell-specific reprogramming may turn out to be one of the greatest strengths of RNA therapies. As suggested earlier, I don't believe it's too controversial to say that cirrhosis likely requires combination therapies targeting different cell types in a highly specific manner, the type of specificity that is difficult to achieve with standard drug regimens. So to conclude, the integration of cell-specific cell reprogramming therapies with machine perfusion and deep phenotyping provides, we believe, one of the most promising new opportunities to make a much-needed impact in patients' lives. With that, I'd like to thank our Oxford, Taipei, and XLab teams and our partners. A special mention to Peter Friend, Rutger Ploch, Hussein Abbas, Carla Cereza, Simon Afford, Heineck Mergenthal, George Clark, and Organox.
Thank you very much to Quin and to all of our speakers for those fantastic presentations. It's now my pleasure to hand over to Dave to introduce the Q&A session. Thank you very much, Dave.
Thanks, Jessica, and thanks very much to Zoltan, Steve, and Quin for their excellent presentations, and also for finding some time over the next 15 minutes or so to answer some questions. Let me just encourage the audience to continue to submit questions, but we will certainly get started with the questions that we have in hand. Let me just make sure that our presenters, Zoltan, Steve, and Quin, have all unmuted themselves and can hear and respond to my questions. Let me do a quick check. Zoltan, are you there?
Yes, I'm here. I hope you can hear me.
Thank you, Zoltan. Steve?
Yep, here, thanks very much.
Thanks, Steve. And Quinn?
Hopefully you can hear this now.
Yes, thank you very much, Quinn. Perfect. Well, for all three of you, thanks again for great presentations. I think there's massive scope for discussion on this topic. I'll try and sort of lead us into that discussion fairly gently at first. But I can see, given the slightly different strategies that the three of you have taken, that I'm sure there's some scope for valid discussion. Let me start perhaps with you, Zoltan, trying to understand how you go about assessing the druggability of the targets that you identify from the in silico screens that you run.
Yes, that's a very good question, and we are, I think, every day this word druggability pops up. So we have different strategies, and it's still work in progress. So we of course try to gather bioinformatic data as much as we can from different databases. There's tractability buckets, and this is something that we incorporated in our automatized target filtering workflow. On the other hand, druggability is not as simple as that, because these databases only consider small molecule inhibition and partially also antibodies, but we know that there are other modalities. So we basically don't discard targets based on bioinformatic data and suggestions, but we then take our targets and we perform computational chemistry analysis. We are talking with different advisors, and then we try to we have a head of chemistry. So we then try to assess the risks of a given target, and we try to also come up with different assays before we actually decide that this target becomes part of our pipeline.
So I know, Steve, this is a topic that has vexed you for quite some time, and I know Engine has spent a lot of time thinking about how you triage targets from the CombiGEM platform. Would you like to say a few words about your strategy in this area?
Yeah, so thanks very much, Dave. Good question. So, you know, obviously, I think the challenge that all groups that are using you know, high throughput screens and computational approaches will identify multiple different hits, and often the challenge is in narrowing that down. We very much take a drug discovery focus to that question. So the question of druggability is obviously a good one. We tend to do it a little differently, I think, from what Zoltan was describing. A, we are primarily focused on small molecules, but B, we tend to assign a druggability score to essentially every gene in the genome. So we have a proprietary way of assessing that that's based on a number of different criteria that include, you know, whether there are known ligands for proteins of interest, whether there are homologies to known druggable targets, whether there have historically in the literature been modulators of those targets. And sometimes we apply that filter upstream because at the end of the day, the targets we're trying to identify we want to develop, and sometimes we can apply that at the end. And there is an advantage to sometimes applying that at the end, because you can get a lot of mechanistic information from targets that you may not be able to develop, but nonetheless it gives you a lot of insight into potential gene interactions that may be helpful in thinking about drug discovery strategies. But in addition to druggability, we're also looking at, as Zoltan talked about, prevalence of the biomarkers and ability to assess the functional relevance of a biomarker. So is it going to be a complete loss of function? Is it going to be a gain of function, etc.? And can you assay for that? And will there be a good diagnostic that you can use in combination with your target? We also look to see that there is strong synthetic lethality. Obviously, the rationale for that is you want to have a very good therapeutic index. You want to be killing just the cancer cells which have the biomarker and not the normal cells which don't have the biomarker mutation. And then we also take into account, based on biomarker prevalence as well as other criteria, what cancer type we are going to pursue, and is there a drug development path there? What does the landscape of therapeutics in that cancer already look like, and how might we fit into that therapeutic landscape? So it's a real combination of multiple different criteria, but I think the common theme here is that we're focused on the drug discovery feasibility.
Thanks a lot, Steve. I think we'll return to that question a little bit later with what looks like a very nice closing question for you to look forward to. But let me turn to Quin now. I'm interested, Quin, you talk about simulating RNA signatures as part of your target discovery platform. Why did you choose that particular strategy?
Well, you know, Dave, I imagine there are really two answers to that. I mean, the first is the question around digitizing biology and in silico screens and the kind of in silico liver we talk about. And then why RNA biology? Well, RNA biology because we're an RNA therapeutics company. So, you know, that's our focus. I think why our particular screening strategy is, I think for us, you know, in the cardiometabolic space, I think we have a slightly tougher time than the cancer space, because when we need to understand biology, their interactions are often very complex in terms of the cell types speaking to each other. And when we're doing screens, you need to, or training up in silico models, you do need to have that complexity. You know, and this is why, for example, we focus on perfusing whole human livers and training our models on that. You know, it's trying to find the right balance, and we'll see where things go. You know, our prediction is sort of by about halfway through next year, we'll have generated enough single-cell sequencing data on human liver tissue that's been perturbed and then single-cell sequenced to really start making quite reliable predictions about phenotypes, so that you don't need to do big brute force screens in simple models. You can go direct to you know, human liver testing and very quickly converge on the right biology.
And Quin, do you think that biology will converge on single targets? I mean, you intimated in your presentation that that might not be the case. So in that scenario, do you envisage combinations of different RNAi therapeutics?
No, definitely. Yeah, the way we break it up at the moment is two levels of interactions. There's within cell and between cell interactions. You know, and of course, the within cell interactions are things like the cancer equivalent is, of course, synthetic lethality, as we've heard about. In our space, slightly different, but the same idea, right? We're trying to find weaknesses in gene networks within a cell. Within the liver, equally important are really the cell-cell interactions. You know, so first part was genes talking to each other. Second part is cells talking to each other. And we know, for example, with cirrhosis, which is the endpoint for so much of chronic liver disease, that it is at least 5 major cell groups contributing to the problem, very actively driving and contributing to the problem. And you know, our feeling is you really do need to be targeting all of those 5 cell types to make a difference.
And I guess the progressive nature of the disease may mean that you need a different combination of those therapeutics at different stages of disease.
That's absolutely true. You know, so for example, with us, you know, we are very focused on the transplant space right now. You know, it's such a global unmet need. I think what a lot of people don't appreciate is, you know, obesity is now becoming the major driver for liver cirrhosis and needing a liver transplant, but equally it's the major driver for the shortage of donor livers. So what we're doing right now is we're focusing on the donor liver challenge, but it's the same target biology. Our hope is that those targets will prove equally good within patients. Yep, it's a thesis. We'll have to see it. It might turn out, as you say, to be slightly different combinations depending on the patient and depending on their stage of disease.
Fantastic. Thanks, Craig. Zoltan, maybe a kind of related question for you. To what extent do you envisage that your in silico platform can predict the responses to known drugs or interventions or even target knockdown in individual patients eventually?
Yeah, this is a very exciting question, and we are indeed thinking about that. And I think this is our goal, really, to build that model based on in vitro simulations, based on training loops and feedbacks, and extending this model to really cover all the proteins in our cell, then become able to really feed in patient data. And I think this is our vision, that once we have such a model, modeling individual patients will be doable, and not only modeling, but also then testing potential drug targets, and maybe within a period of a few days, we will be able to predict which drug the patient will react to. And I think this will benefit everybody a lot.
Fantastic. And for you, are you mostly focused on small molecule modalities, or do you envisage also getting involved in interference RNA or other modalities?
We are open. We always keep in mind other modalities. We also have collaboration partners regarding modalities apart from small molecule. We have now currently some targets in our pipeline for what we are focusing on small molecules. But like I said, we are very open to other modalities as well.
Okay, thanks. Steve, you briefly mentioned applying your platform outside of the oncology space. I wonder, which therapeutic area do you think is most tractable and most attractive to use this strategy outside of oncology?
It's a good question. The technology itself is not particularly therapeutic area limited, and in fact, we already have had successful collaborations in other areas, in particular in dermatology, working with Procter & Gamble, where we were able to identify, in that case, combination therapies that might be particularly useful in skin disorders. But one of the areas that we're particularly interested in is neurodegenerative disease. And I think, one of the reasons for that is that there's a huge unmet medical need. We're really, in desperate need of new and effective targets. And yet, the raw material for the types of approach that we're taking, that is to say, genomics data, is abundantly available. There are large numbers of collaborations and consortia that are generating data across neurodegenerative disease. And we can apply technologies that we are applying in oncology, with the important addition of being able nowadays to be able to actually look in patient-derived neurons by virtue of using iPSC-derived neurons from patients. So our technology and the CombiGEM technology can be applied in iPSC-derived neurons, and therefore be able to answer questions about what particular targets may be particularly suitable in neurodegenerative disease. So we're excited to pursue that area.
That's great. Thanks, Steve. We're almost out of time. I'm going to pose one last question, same question to all three of you. And we only have time for a very, very brief response, I'm afraid. But my question is, what do you think are the main obstacles to opening up traditional drug discovery pipelines to big data and AI-based approaches right now? Let me start with Quin.
Skills, that's for sure. Molecular biologists don't really have enough overlap with computational biologists in our space right now.
Great, thank you, Quin. Steve, what, in your view?
Yeah, I think that's a good response. I think that one of the things that we do at Engine is to really integrate our bioinformatics groups with the molecular biologists and drug discovery teams, and I think that is very productive. That the whole field of network biology is really has been spawned from the interface between computational bioinformatics and molecular biology. So that's an important consideration and something that's really developing.
Thanks, Steve. And Zoltan, would you add anything to those responses?
Yeah, it's a very good question. So I think the availability of good quality data is crucial, and I think we have a lot of data around. But really, to have trustworthy data that can be used for training is, I think, very important. And I believe that a good algorithm is great, but having a lot of data for training is even better.
Great, thanks a lot, Zoltan. So we're at the hour. I'd like to finish by thanking again our three presenters, Zoltan, Steve, and Quin, and thanking the organisers, Oxford Global, for hosting us, and thanking all of our attendees. A recording of this webinar will be made available, and you'll get an email providing a link to that. So thank you very much, everybody, for your time today. I hope you enjoyed the presentations and discussion. Goodbye.
Thank you very much.
Thank you very much. Goodbye.